Thank you for joining us today. In this talk, I'll be discussing surface fluctuating hydrodynamics. So my work is motivated by a number of different application areas. I don't have a lot of time to go into this, but I've been doing work on modeling proteins and their diffusivity within lipid bilayer membranes. And one of the things that we're particularly interested in is accounting for not only the hydrodynamic coupling within the membrane, but also in the roles that geometry plays. On the upper right, I'm showing vesicle shape changes that are induced by proteins or other chemical species that are immersed within the membrane, which can change the local spontaneous curvature. The key point I want to make there is that it's not only important to take into account the elastic mechanics and the hydrodynamic coupling, but also the equilibrium shapes in these two different states are often separated by energy barriers, and so thermal fluctuations play an important role. And in the bottom, I'm showing some work with Patricia Basseur that we did on modeling the interactions of proteins with membranes and the role of geometry in single particle tracking experiments. So in this talk, I'm going to talk about computational methods by which we can start to incorporate geometric effects into models of hydrodynamics and also incorporate in a self-consistent way thermal fluctuations. So there's, of course, a lot of different areas that this type of issue arises. And really the key challenge is coming up with a tractable description of the hydrodynamics as well as efficient computational methods. The problem area that we've been particularly motivated by, as I mentioned before, was protein interactions within lipid bilayer membranes. So you can think of a protein as being effectively like a little cylinder immersed in a slab of fluid. And what's interesting, and this work goes back to Safman and Delbruck, is that you might be tempted to model the membrane as just a two-dimensional fluid sheet, but you very quickly realize that there's not a well-defined mobility, and by extension, by the Stokes-Einstein relations, diffusivities in the two-dimensional setting because of the Stokes paradox. And so what was realized is that you need to take into account the role of the surrounding solvent for the solvent. If you take that, Safman and Delbruck worked out in the 1970s that in the asymptotic limit of an infinitely thin sheet, you actually find that the mobility response depends logarithmically on the protein size. And this important length scale, which is known as the Safman-Delbruck length scale, which is the ratio of the membrane viscosity to the solvent viscosity. So this is quite interesting, and in contrast to what you would see in bulk three-dimensional fluids, where the mobility response would scale as 1 over the size of the protein. So this also has other interesting implications because the flow structures that you end up with in the membrane setting turn out to be far larger than you would have in bulk three-dimensional fluids. And so you see even for a small protein on the order of 10 nanometers, this Safman-Delbruck length is typically on the order of a micron, and therefore you get flow structures that are approaching the length scales on which you get interesting changes in the geometry from curvature or potentially even from topology. So I'm going to talk a little bit about our strategy to do that, leveraging results from exterior calculus. The main motivation here is to be able to generalize the types of operators that arise in continuum mechanics to the manifold setting effectively. And we'll do that using the exterior derivative, Hodge star and wedge product from differential geometry, which I've summarized over here on the right. And this very nicely allows us to express things like the gradient, divergence, and curl in terms of a combination of these operators on the surface. And I'll also mention that we'll formulate the mechanics in terms of the cotangent space, using these flat and sharp isomorphisms to move back and forth between vectors and their duals. I'll just also point out over here that you can express important tensors, like for example here, the divergence of the rate of deformation tensor on the surface in terms of these operators, and this would be effectively the contribution of the Newtonian stresses in the interface. So one can use the Stokes theorems to derive effectively conservation laws, as you would in traditional continuum mechanics, but now taking into account these contributions of geometry. And so here I'm just showing conservation of mass equation, where we've used Fick's law as the constitutive relation to operators. So we can do something similar to generalize things like the Rayleigh transport theorem and so on to derive our fluid mechanics. And so we've done that, and this is what we obtain. One of the challenges that comes up in the manifold setting is imposing the incompressibility constraints. The way that we're going to approach this is to generalize the idea of vector potentials to the manifold setting, and so we'll use this generalized curl of a scalar field. So we're able to exploit the Hodge decomposition as you have in the Euclidean setting. Okay, so the price that you pay by using this Hodge decomposition approach with this vector potential is dealing with a fourth order operator, as I've shown here. So focusing on the Stokes flow, I'm going to just briefly discuss how we would model a lipid vesicle. In particular, we're going to model each of the leaflets. We have an inner leaflet and an outer leaflet. we're going to model the intra-monolayer lipid flow, the intermonolayer slip between the two leaflets, and we'll model the traction stress with a bulk solvent fluid. So our model is effectively going to consist of 4 fluids. We'll have a fluid trapped in the interior, we'll have a fluid shell for the inner leaflet, we'll have a fluid shell for the outer leaflet, and we'll have a bulk surrounding the 3-dimensional fluid. And so these are the equations that go with that, and it's effectively the flow equations that I showed you on the previous slide. The traction stress is denoted here with the interior and external fluid, depending on the leaflet, and this is the intermonolayer slip at that there. And then we have, of course, any body forces, and we have the pressure to impose the incompressibility. Now, to get the contributions of the flow outside of the membrane, we'll solve these Stokes equations on the right. I'll just mention there's an interesting asymmetry here in that the flow on the interior fluid here has an interesting non-dissipative mode where it can undergo rigid body rotations versus the outside fluid, which we're assuming it's rest at infinity. So to handle the proteins that are immersed in the bilayer, what we're going to do is take an immerse boundary approach. And if you're not familiar with the immerse boundary method, the idea is that I can model microstructures by essentially putting markers in my fluid. I'm going to advect those markers along using this velocity averaging operator, which I've denoted by gamma here. And any forces that may act on the body, I'm going to account for those by spreading those out as force densities in the fluid equation, given by the capital lambda operator here. And now what's really interesting is that if you want this interconversion to not be lossy, in other words, the coupling to be non-dissipative, you can write down the energy equations, and what you'll find is that that requirement results in an adjoint condition between the operator gamma and lambda. And so what's nice about that from a modeling perspective is that if I come up with a velocity averaging operator, this allows me to obtain a corresponding force spreading operator by simply computing the corresponding requirements in the adjoint condition. So that's what I've done here, and the way we're going to do this in the manifold setting is we've come up with these probing vector fields which get us the velocity response in each direction, weighted of course by the distance relative to the protein inclusion. And so we can create probing vector fields both for translational and rotational motions, as I've shown up here in those green fields. Now the details, I'll refer you to our recent paper in 2019, and you can also look at our 2016 paper for a full explanation of this. And so we can use this to compute mobility responses by effectively doing force spreading, doing a fluid solve, and velocity averaging. And so now we can get the translational and rotational motions of a large collection of proteins within the bilayer in this manner. So the main challenge then is coming up with a good fluid solver for that vesicle model. Our approach is going to be to develop spectral methods with Galerkin truncations. And so the key to doing that is going to be able to compute this inner product between any target force density acting on the membrane and the corresponding spherical harmonic mode. And so the way we're going to do this is we're going to expand our surface vector potential in spherical harmonics, and then we're going to similarly do that for the body force acting on the membrane. Now that part needs to be done numerically. You might be tempted to use latitude and longitude coordinates, but we find that that's problematic with the immerse boundary method because you get sort of different clustering or density of sampling at the equator versus the poles. And that leads to kind of inconveniences and how you would have to sort of add additional contributions in your immerse boundary method to kind of compensate for that in your weight functions. So instead what we've chosen to do is to use an approach that's based on Lebedev quadratures. The other thing I'll mention is that the spherical harmonics modes are in fact eigenfunctions of these equations, and this allows us to proceed quite far analytically without having to do any numerical calculations in formulating the equations of the response. And so here what this shows is that we can basically decompose these two equations into a bunch of 2x2 systems for each degree of the spherical harmonics. You also see an asymmetry here from the traction stresses of the inner leaflet and the outer leaflet there, given that rotational mode. Okay, so to do the inner products that require the key piece of information, which is the load force density on the surface, we use the Lebedev quadrature weights that are almost uniformly distributed on the surface. These are designed to be accurate up to 110 degree spherical harmonics, and they have octahedral symmetry. And so this works quite nicely in practice. Here's an example flow field we've calculated using this technique. You can see that we have a flow force here at the North Pole and symmetrically on the other side, and you can see the flow recirculates quite globally over the surface of the sphere. You can use this to compute collective dynamics, and the hydrodynamic coupling has some interesting implications for how proteins sort of couple within the leaflet. Because of that incompressibility, as proteins come together, they kind of squeeze fluid out, and you can end up with very interesting collective diffusion effects as a consequence of that. But we've done a number of studies using this approach with active inclusions. I'm just showing a few here. Let me just make a few remarks about the interplay between the hydrodynamics and the geometry. So we can do non-spherical shapes as well using this spectral approach based on spherical harmonics. The idea is very similar to what I outlined before, except we can't analytically decouple the system into 2x2 blocks. Now instead we just compute a global stiffness matrix by using the Galerkin truncation and then solve those set of system of equations. So this shows the hydrodynamic response with these three load forces. And one of the interesting things that we find is that the flow structure can significantly change when the geometry of the manifold changes. So here you see the creation of these new vortices and the flow tending to want to recirculate more locally. And you see a creation of a saddle point there. And that's similarly for this other more complicated shape. And so that's quite interesting because we get a significantly different arrangement of the flow on the surface for comparable load forces when the only change here really has been the overall geometry of the surface. So these variations in curvature seem to lead to additional sources of dissipation within the flow. And so what we did to study this more quantitatively is we looked at the Rayleigh dissipation principle, which serves as a variational principle for Stokes flows. In particular, Stokes flows minimize the Rayleigh dissipation. And what we decided to do is look at what the push forward looks like of the flow on the sphere to these shapes, and to compare that with the Stokes flows that we obtained from solving the hydrodynamic equations. And you can see that these local recirculations lead to significant savings in terms of the Rayleigh dissipation relative to the push forward flows. And so that's quite interesting, and that could have implications for, again, how proteins would interact within a bilayer if there's some interesting local curvature in terms of how their collective dynamics evolve. And so this is something we're studying a little bit more detail, and I'd be happy to talk more about that if that's of interest to people. Thermal fluctuations, let me discuss how we are incorporating that into these hydrodynamic computational methods. In particular, we're modeling the fluctuating hydrodynamic in an inertial regime, where we have both the fluid equations and the momentum equations for the microstructures. And these are going to be coupled using this local drag law, where essentially we're going to have a Langevin equation for the microstructures, but with the twist that instead of referencing a zero background, we're now going to reference a velocity averaged value that we get from the local flow environment. So we'll use the velocity averaging operator gamma to obtain that value. And then because the drag on the particle, there's an equal and opposite force on the fluid, we'll change the sign here, as you can see, and we'll use a force spreading operator to account for that loss of momentum from the fluid as it drags on the particle and vice versa. Now, what's interesting here is how do we thermostat this system in a way consistent with statistical mechanics? And the way we do that is we use the fluctuation dissipation principle, which means that basically the thermal fluctuations, this field here and here, will depend on the dissipative operators of the system. And so the force densities that we get for the thermal forcing of the fluid equations is given by this equation here with this dissipative operator LFF. For the microstructure equations, we get something similar to the Langevin dynamics with drag gamma. And then these two fields are actually coupled to one another. And that's occurring because, again, there are certain parts of the momentum, in particular through this drag, which when they're lost from the fluid, they're gained by the microstructure and vice versa. So in some sense, the stochastic forcing terms, part of their role will just be exchanging momentum between the microstructures and the fluid. And that's manifested by this cross correlation. Okay, so the dissipative operator for the fluid is given by the usual surface flow. This is manifested here from the Newtonian stress of the surface flow, the traction stress here, and then this is from the drag term here. And so you can see that you get some pretty interesting results from the fluctuating hydrodynamics. You get these long-range correlations, which are manifesting from the incompressibility constraint. You get some interesting interactions as well with the local geometry of the surface. We validated this with respect to statistical mechanics, and we've also done derivations to get this in the overdamped regime. And so you start to see that there's interesting phenomenology that occurs because of the hydrodynamic coupling. Here you can see the pair meeting times have a significantly longer tail when there's hydrodynamic coupling versus the non-hydrodynamic case. I think I'll skip this slide in the interest of time. We've been looking at different kinetic processes within membranes with and without hydrodynamics. the thing I want to talk about for a few minutes is the velocity autocorrelations and how they significantly differ with bulk three-dimensional fluids, which has a power law of t to the negative 3 halves. We see that there's these different regimes for surface fluctuating hydrodynamics, where when the vesicle is very big, what's going to happen is we get a t to the negative 2 power law. And this occurs because the velocity decorrelation essentially is being dominated by the time scale it takes for shear stresses to kind of propagate over the surface of the vesicle. But as the vesicle becomes smaller and smaller and smaller, what happens is that the shear stresses can dissipate over the entire vesicle very rapidly. And if that occurs very rapidly relative to the time scale of the traction dissipation, you can end up with this plateau regime. And then, of course, then the traction, the rotation of the vesicle dissipates the rest of the momentum. And so what you end up with here is these two time scales, tau f for the in-plane shear stress and the tau r for the rotational time scale. And you can see that when there's a gap between those two, you can get some pretty interesting phenomena in the velocity autocorrelations. So actually, the size of this vesicle would play a significant role in the type of velocity autocorrelation behavior that you would obtain. And so that differs from the three-dimensional bulk, obviously, not only in the quantitative scaling, but also in some of these qualitative properties. So again, there could be some pretty interesting implications, especially if using this type of description to look at scattering data sets and things that can resolve things at this level of the dynamics. So we did also studies with the Galistenian swimmer, as I mentioned before, and what we in particular looked at was how having active entities within the membrane could potentially enhance mixing or effectively the diffusivity. And so we find, as a function of the number of these Galistenian swimmers, that you can get a factor of 1.5 all the way up to 2 here in terms of the effective diffusivity, enhanced diffusivity relative to the bare thermal diffusivity. And so again, this is kind of a proxy for mechanistically thinking a little bit about what active conformational changes of proteins might do within lipid bilayer membranes. So we've also been working on developing other types of solvers to allow us to go beyond just spherical shapes into other topologies or more complicated shapes. And so the idea of this generalized moving least squared approach that we've been developing is to represent the manifold as a point cloud, and then if there's a target operator that we want to approximate at a point xi of some surface field u, the way we do that is we have a least squares problem that we solve basically to reconstruct a proxy for u, where these lambda i's are probing functionals that kind of tell us what important information to pay attention to. Now, typically, these will simply be point evaluations of u, but you can also put in there moment conditions that could be relevant to your particular problem or properties that you're trying to preserve. And then the other novel part of this is that instead of the least squares problem being global, you basically weight the data according to how far you are from that target location xi. So now that you take p star, you can use that to get an approximation to your target operator by evaluating the target operator on the reconstruction p star. And so what's interesting here is we're not only doing this for the differential operators, but because the differential operators depend on geometry, we also have to do a separate stage where we estimate geometric quantities on the surface. So this is just showing we can do this by creating a local sort of tangent plane by doing a PCA on the point cloud representation of the manifold, do a local Monge gauge, and then from there we can compute things like the local Gaussian curvature. And this is just showing one of our studies that is able to capture that to fifth order accuracy. So we also can do this to solve this bioharmonic equation for the surface vector potential, and we've done that as well. This involves a little bit more stages, and there's a few more target operators involved. You take the surface force density, convert it to the corresponding one form. You use GMLS to do a generalized curl to get it into the form you need on the right-hand side. You can formulate a collocation matrix for that bioharmonic equation, solve that, and then once you've solved that, you can get back the vector field by doing, again, another generalized curl, and then by doing a sharp, you recover the surface velocity field. So in this way, we're able to use these techniques to solve flow fields on quite general geometries. And this is just showing a few examples here on the right, and we're also working on doing this beyond spherical topologies, where you would also solve a separate equation involving the harmonic functions. So here is just showing that we have pushed these methods up to solvers that can get sixth order accuracy. So I'll refer you to this paper down here for more details. So I think that's a good place for me to conclude. So I hope this has provided some overview of the work that we're doing on extending Safman-Delbrück type results for capturing collective hydrodynamics in curved fluid interfaces. Also, I hope to give you some indication of how this exterior calculus from differential geometry can be used to develop both analytic techniques for hydrodynamics on manifolds as well as efficient solvers. And also, I hope it has given you some exposure to this surface fluctuating hydrodynamics that we've introduced for drift diffusion dynamics of microstructures. So I think there's a lot of potentially interesting application areas, and I'd be very happy to talk further with you if this is of interest to use. I'll refer you to the papers here in the meantime, but feel free to see our website and to contact us. I would enjoy continuing the discussions. I wish we all could be there interacting in person, and hope to see everybody soon.